Alright, this is Unit 2 of Biochemistry 1. There's some good reading on the first couple slides. I'll let you do that on your own. The only thing on this one is to not worry about these numbers. Don't waste your time studying any of this stuff. What you do need to know is about hydrogen bonding, and you're always going to be looking for OH or NH mainly for hydrogen bonding. SH is another one that will cause hydrogen bonding, and carbon and oxygen, there'll be some polarity in this bond, so there'll be potential for hydrogen bonding as well. But those are the main things you're going to be looking for, the main types of bond. It's another very important slide to know the weak interactions and the definitions. And on this slide, definitely know these orders. So, covalent or strong interactions, here are your weak interactions. So, hydrogen bonding will often be referred to as the third strongest or the third weakest interaction, because it's right in the middle. That's one of his favorite things to do. So, definitely know these in order. There are two slides, one on slide 17 here and another one on slide 26, that are talking about the properties of water. Those will almost be guaranteed to be on the tests and quizzes, so make sure you have these properties of water down really well. We'll come back to sodium dissolving in water. This is an important slide. It usually shows up. So this will be written as N2 or O2, CO2, NH3 or SH2. So the more polarity there is, the more soluble it is. So hydrogen sulfide has the strongest polarity, therefore it is the most soluble. The more bonds you have, so there's no polarity here, plus a triple bond, it's the least soluble. This is the other one I was talking about, properties of water. Make sure you know this. Water is both a nucleophile and an electrophile. This is kind of a organic chemistry review. Definitely be familiar with all your functional groups again, and really know examples of where they're found. For example, phosphodiester's, he won't have nucleic acids, he'll have DNA or RNA, for example. He won't say proteins, he might say insulin. If you don't know what it is, it's probably a protein. All the other ones are pretty obvious. Carbohydrates, it'll be something like glucose or fructose, sucrose, will end in a ose. So proteins is the one to really look out for there. This is showing proof that the equilibrium constant for water is 1 times 10 to the negative 14. Very important number to know. This is some acid-base chemistry review. So there's a few equations you're definitely going to have to know. The pH equation is one of them. I'll point them out as we go along. The list of strong acids and strong bases, make sure you know those. One to note are the numbers here, so HClO3 is weak, or HSO4, that's weak, but these are the strong ones. Another one that he likes to throw in is HNO2. That's another. So these are all weak ones that look very similar to the strong ones. Just be on alert for that. Right, so this is another equation you have to know. I don't like that form of the equation. It's very confusing to me. The way I learned it in chemistry, and I still used it for all of his problems, is Ka equals the concentration of hydrogen squared, don't forget the squared, over the concentration of HA, or the initial concentration. This is the equation I use. Remember, this is for weak acids. And you're usually going to be using this one in combination with pH equals the negative log of hydrogen. So this right here is how you relate the two equations. So we'll talk about the math in SI. The problems are pretty self-explanatory. We'll show examples of them. Don't worry about memorizing all these weak acids. Just know that carboxylic acids are weak unless otherwise stated. There's only one he'll give in Unit 3 that's a strong acid that's carboxylic. Pretty much the same thing with bases, except the inverse. This is a big source of questions, so make sure you understand this slide, that all salts will dissociate completely. If it's neutral salts, so two strong ions, usually from a group 1 and a group 7 combined, like NaCl or maybe potassium bromide or something, those are going to dissociate completely, will not affect the pH. If you have something that ends in 8, so this is sodium acetate, I believe. If it ends in 8, it's a weak ion and it's going to be basic. So after this equation, the product is you're going to have an increase in pH. Ammonium, NH4 plus, is what he's going to use almost every time for the acidic example. Ammonium NH4 plus will release that extra hydrogen, causing the pH to go down. So really make sure you understand this slide. This buffering regions are going to be summarized on this slide here, 55. So if the pH is 4.76, you want a acid-base solution, so say an acetic acid acetate solution of 4.76. That's the best. 5.76 or 3.76, those are the good range. Anything else won't really work as far as a buffer goes. It really needs to be within 1 point, ideally, of your original pH that you're trying to keep it at. So I went back a few slides. I'm back on slide 47 now to kind of illustrate what I was just saying. So our solution was at 4.76, so we can have a range 1 above or 1 below, and that's the range you can buffer in. Anything beyond, it goes out of control. Some calculation examples. Again, don't worry about memorizing any of these numbers or anything. The pKa's will always be given for these problems. On this slide here, CO2 or HCO3 bicarbonate, those are going to be blood buffers. And the pKa is about 6.4, and the blood pH is 7.4, so it's 1 point away, so it's still a good buffer. The other thing to know is phosphate. So right here, the pKa of 6.86, this is buffering the cells. And the cells have a pH of somewhere between 6.8 and 6.9, depends. So associate phosphate with cells and CO2 and bicarbonate with the blood. I would know those numbers. Anytime you're working with a buffer solution, you're using the Henderson-Hasselbalch equation. This is another one you have to memorize. So it's pH equals the pKa plus the log of base over acid. Always base over acid. And that can be in moles or molarity. As long as they're both the same, it's a ratio. So most of the time you're going to get it in molarity, but in the labs you might get it in moles. it's okay, as long as they're both in the same unit. It's all about the ratio. So again, CO2 buffering blood. Don't worry too much about the reaction, just recognize that all three of these forms are part of the CO2 blood buffering. So you can see bicarbonate written out like this or this. CO2 could also be an option for carbon dioxide. So here's just an example using the Henderson-Hasselbalch equation, showing another example using the cell buffering phosphate. And this is leading into the next unit, but also know that proteins are very good at buffering both blood and cells. And that's all for Unit 2.